Today, the goals of this lecture are, we'll do a bit of recap, we will talk about drugs, we will try to define drugs, then we'll try to talk about broad mechanism of action of drugs. Then we'll try to look at structural features of protein receptors. We'll talk about the broad classification of drugs. We'll talk about side effects. And we will end with the discussion of some characteristics of small molecules, small molecule drugs. So this is our goal for this lecture. So as a recap, we talked about how do we get new drugs, meaning what strategies are adopted. So what strategies are adopted? So we talked about that we get drugs from target-based screening, we get drugs from phenotypic approaches, we get drugs by modifying structures of natural products. Natural products meaning drugs which are obtained from living organisms, which may be plants, which may be animals, which may be marine organisms, etc. And we talked one of the strategies that is adopted to get new drugs is a follower drug strategy. Okay, this is also called, we try to make a best-in-class. Best-in-class drugs, meaning a class already exists, and we try to add value to existing class of drugs. Okay, what value are we talking about? Maybe it has got an increased duration of action, or maybe the number of times it has to be given in a particular day is reduced. You have a TID drug, which is the existing class, and you make it into a OD once in a day. So that becomes the best in class. So that's a follower drug strategy. The other one is biologics. Okay, biologic based approaches. There is one other one, that is drug repurposing. So that is a drug which is already licensed for use for a particular therapeutic condition, and now we have a new indication for that same drug. So this is called drug repurposing. So these are the strategies currently which are being followed to get drugs, and these are the aspects that we alluded to in an earlier lecture. So, what is a drug? To answer this question, let us pull up the browser. And let's type the Drugs and Cosmetics Act 1940. Let's pull up an official document. Let's save it on the desktop. Okay, so we have saved up this. Let's come back to, let's transition back. And let's pull up that file. Okay, let's see which page are we looking at. Okay, let's pull up this page. Okay, so we are trying to define what is a drug. So for that we have pulled up the official copy of the Drugs and Cosmetics Act 1940. And this Drugs and Cosmetics Act 1940 is of the country of India. So this is a legal document that defines what a drug is. Let's look at what the document says. So these are the definitions given for a drug. So this is the legal definition of a drug. Let's read through this. All medicines for internal or external use of human beings or animals and all substances intended to be used for or in the diagnosis, treatment, mitigation or prevention of any disease or disorder in human beings or animals, including preparations applied on human body for the purpose of repelling insects like mosquitoes. Okay. And there are definitions which broaden the scope of the first paragraph. And that is the legal definition for a drug as far as we are concerned. Okay. So for us, drug a drug in the last one of the previous lectures we have talked that a drug has got a mechanism of action. We said it produces its effect by acting on something called the receptor, and that results in a therapeutic response. Okay, so a drug acts on something, and we said that something is called a receptor. And then we went on to say receptors generally could be proteins, or they could be nucleic acids. And we went on to talk about lots of different examples. Okay, we said proteins could be ion channels, they could be transport proteins, they could be enzymes, they could be structural proteins, so proteins could be any of more than all these types of all these are types of receptors: ion channels, transport proteins, enzymes, structural proteins, to show a few. Then we said some receptors could be nucleic acid, but the majority of the receptors, so the majority of receptors are proteins. So the majority of receptors are proteins, and therefore we need to learn about proteins. Okay, because the majority of receptors are proteins, we need to learn about proteins. Learn about proteins. So what are proteins? So in the last lecture, we said proteins are macromolecules. Okay. So in case of proteins, they are made up of simpler units. Okay, so they are made up of simpler units, and these simpler units could be thought of as being monomers. But these monomers come in 20 different flavors. Okay. So the simple units, we call them amino acids. So these amino acids come in different flavors. There are 20 amino acids, and combinations of these amino acids give us proteins. Okay. So let's say there is an amino acid and another amino acid, another amino acid, another amino acid. So chains of amino acids. Okay, so this is a chain of amino acids. And when amino acids combine with another amino acid, then they do so through what is called a peptide bond. So all these are basically peptide bonds between the amino acids. These are all peptide bonds. Peptide bonds. So now there are 1, 2, 3, 4, and 5 amino acids. So we call this a polypeptide. Okay, meaning there is more than one peptide bond, so we call it a polypeptide. Okay. So, peptides are defined as having 2 to 50 amino acids. Okay, so if there are 2 to 50 amino acids, we call it a peptide. So each amino acid is called a residue. Okay, so now we say here there are 5 residues. 5 residues in this peptide chain. Okay, so now we know what is a residue. Okay, so we said a peptide contains 2 to 50 amino acids. Now, these peptides are divided. They can be polypeptides, okay. They can be oligopeptides, okay. So oligopeptides, we say oligopeptides contain about about 2 to 20 amino acids. Okay, 2 to 20 amino acids, they are called oligopeptides. And then what is a polypeptide? A polypeptide will contain about 20 to 50 amino acids. Okay, so that's a polypeptide. Okay, it contains between 20 to 50 amino acids. We call it a polypeptide. Okay. Yeah. So greater than 100, we call it a protein. So theoretically, anything greater than, so let's say anything greater than 50, we define as a protein. Okay. So now we know the difference between, we have learned the difference between amino acids. We now know what is an oligopeptide. We know what is a polypeptide. And we know the definition of a protein, okay, in terms of the number of residues. Okay, so based on the number of residues, defined oligopeptides, polypeptides and proteins. So what are amino acids? So amino acids would have a structure like this, and I have drawn the simplest amino acid. Okay, so the simplest amino acid is, we know, glycine. So we simplest amino acid is glycine. Okay, all the amino acids will have a general formula. The general formula would be how can we write the general formula? This is an R group. So this is the general formula of amino acids, where the R moiety would be different in different amino acids. So how can we write this properly? We can say the general formula would be R CH NH2. Okay, so this would be the general formula of amino acids. Proteins, we define proteins as being having residues greater than 50 amino acids, but that is not the whole definition of a protein. A protein, in addition, should have at least primary, secondary, or tertiary structure. Okay, in addition to having residues, it should have a primary, secondary, tertiary, sometimes a quaternary structure. Then we call it a protein. So now we are going to talk about what are these different structures that we are talking about. So let's say the primary structure. So the primary structure of a protein is what we drew initially. Amino acid, amino acid, amino acid, amino acid. So there's amino acids in sequence, it's kind of linear. So this linear chain of amino acids where the individual amino acids are linked by the peptide bond, constitute the primary structure. So then what is the secondary structure? So secondary structure is if this linear structure folds upon itself, okay, folds upon itself, maybe like that, and where the individual amino acids are stabilized by hydrogen bonds, this chain is stabilized by hydrogen bonds. So this interaction or the hydrogen bonds which result in the structure being folded give rise to what is called the secondary structure. So we can have structures which are helices. Say it's an alpha. Let's modify this. Alpha helix is right-handed helix. So that could be an alpha helix, or we can have, so amino acids, when they join together, they'll have an N-terminal end, and that will have a C-terminal end, and sometimes if these amino acids line up and then they are being stabilized by hydrogen bonds, then that is called a beta sheet. So tertiary structure would be when there is interactions between the amino acids which are separated by a long distance. So when there is, say, the molecule folds up upon itself, and that end comes and kind of folds up something like that, where there is hydrogen bond interactions among the chains, among the amino acids in the chain, and where there is interactions like van der Waals. Or there is interaction with water molecules. Then those structures that are formed, they are called tertiary structures. So quaternary structures are formed when different subunits, when a protein has got more than one subunit, and it's folded in a complicated manner, that gives rise to a quaternary structure. Okay. So in the quaternary structure, there are a few things that we need to know. So if a quaternary structure of a protein is taken, and let's say we've got an alpha helix like that, and you have a beta sheet, and again this goes, folds another fold, and comes back like that, and then goes like that. Then what we observe here is these are all stabilized by hydrogen bonds. Okay, hydrogen bonds stabilize this. The structure is maintained by some interactions like that. Okay. And suppose the chain is broken here. Okay? If the chain is broken here, and let us assume this piece is removed, then what would happen is this structure may lose its shape. Okay, it may be unfolded. It may be unfolded. So the removal of this particular segment would change its structure drastically. So we know for a function, so structure is essential for function. So if this structure is lost, then function is lost. Okay? So such groupings of molecules with a particular shape within the overall protein structure, this is called a motif. Okay. So a motif is basically a structural unit within a protein which has got a certain function, but it cannot have an independent existence outside the protein. So that is called a motif. In some proteins, so suppose this is a protein and this is again a piece that has got a particular function. It has got a particular function. Now, if we cut this molecule there and remove it, it will still preserve its structure and therefore its function. Okay, to preserve its structure and therefore its function. So this subunit is called a domain. Okay. It is called a domain. Okay. So let us recapitulate. We said drugs act on something which we call receptors, and these receptors are proteins. Okay, so the drug modulates the protein. So the question is, will drugs modulate all proteins? Will drugs modulate all proteins? So an ancillary question would be, are all proteins receptors for drugs? The answer is no. Okay. So all proteins are not receptors for drugs. So then we ask the questions. So there are proteins which are not receptors, and there are proteins which are receptors. So what is the difference? Okay. Why is that? Some drugs modulate only some proteins. What is this difference? This difference is what we term the druggability. Okay. So druggability is the ability is the ability of a small molecule to modulate a target. Okay. In this case, our target is a protein target. Okay. So some drugs can modulate some proteins. All the proteins cannot be modulated by the same drug. So how do we explain this? Okay, so that is our goal now. We are going to explain what makes a protein target druggable. Okay, what makes a protein target druggable? So let's go back to our browser. Okay, let's go to a software called PyMore. Okay, and then we will take our discussion from there. Okay, so this is a PyMore software. So let us ask it to fetch us XBB. Okay, so that is the code for protein, a kinase on which the drug Gleevec acts. Let's pull it up. Okay, so let us hide everything and let us show it as a cartoon. Okay, and then let's color it as a spectrum. Okay, so this is the structure of a kinase molecule. Now, what is important here to know is these are the helices that we talked about, the secondary structure, and this is the primary structure, which is the chain of amino acids, and this here is the beta fold. Okay, so we are seeing the different structures of the protein. So we are seeing a primary structure. This is the primary structure. This and this is the secondary structure. And the interactions among all of them bring about this whole structure, which is the tertiary structure of the protein. Okay, so this is a kinase. Now this kinase is inhibited by a drug which we call Gleevec. So let us show that particular. So let's show it as a spears. Okay. So this spears is the drug molecule that actually inhibits this kinase molecule. Okay. So we said all proteins are not receptors. This is a receptor protein which binds a drug. So what is the characteristic of this protein that makes it different from proteins that don't interact with drugs? So if we show this as a surface, then we see that the requirement for a drug binding protein is that it should have a cleft. It should have a binding site. Okay, so what is a cleft? It is a region, a fold within a protein. Okay, where the molecule can come, in this case Gleevec, it can come, and this is a kinase. What does a kinase do? It binds ATP, and this is the ATP binding site. So in that ATP binding site, we have put this inhibitor, which is Gleevec, and that drug is inhibiting the activity of this particular kinase. So the point to remember here is drug targets should have a structural feature called folds. Only proteins containing folds which can be reached by a small molecule are drug targets. Okay, so hope that is clear from this particular animation that we see here on PyMo. So let us transition back. Okay, so now what did we learn? What makes a protein target druggable is that the protein should have folds. Okay, and that folds make the protein druggable. Okay, so those are the requirements or the structural requirements. Structural requirement of a receptor. Okay. So the fold of the protein, this is the protein, the fold is called a cleft. So in this cleft, if we add a molecule, that molecule is going to interact with the amino acids which surround the cleft. Okay, one or more amino acids here is going to interact with the drug molecule. Okay, and that is basically the binding of the drug. So when we say binding, we usually mean these interactions, which are usually hydrogen bonds. Okay, hydrogen bonds or non-covalent interactions. Okay, so when the drug reaches the cleft, there is non-covalent interaction with one or more amino acids which are near the cleft, and that is responsible for the drug action. Okay, so we have discussed the structural requirements of a receptor by looking at an example. Which example? 1XBB. Okay, it is a kinase which binds the drug, the trait name of which is Gleevec. Okay. Okay. So now we come to the broad classification of drugs. Okay. So in the broad classification of drugs, we classify drugs as small molecules and large molecules. Okay, so what is the threshold? 1000 Dalton. So below 1000 Dalton, it is a small molecule, and greater than 1000 Dalton is a large molecule. These large molecules, they are also called biologicals or biopharmaceuticals. They are also called biotechnology, derived drugs, So the companies which produce small molecules, they are called pharmaceutical companies. They are also called pharma companies. The drugs that produce biologicals, they are called biotech companies. Okay. All right, so now let's talk about side effects. So the concept to remember is that the Paul Ehrlich, his concept was a drug binds to a receptor, it forms a drug-receptor complex, and this drug-receptor complex brings about the effect, and that effect translates to a therapeutic effect. The underlying assumption is one drug binds to one receptor. Okay, so this is the underlying assumption. This is desired in an ideal world, but in a cell, this is not strictly true. So what happens is, practically what we observe is this drug binds to many receptors. Okay? So the receptor to which the drug is supposed to bind the main one is called the target. Okay, but it binds to many other receptors. So those receptors then are not the target, so they are called off-target. Okay, by off-target we mean outside the target. Outside the target. Okay, so these off-target effects are responsible for the side effects that are observed with drugs. Okay. So now let us look at what are the features that are expected of a small molecule drug. Okay, so in this discussion, we are talking only about small molecules. So the question is, what are the characteristics? Characteristics of small molecule drugs. Okay, we have already mentioned the first one, which is that it should have a molecular weight less than 1000 daltons, and that we called it the definition of a small molecule drug. But it was observed a big data set of low molecular weight drugs were studied, and it was observed by Lipinski that among the small molecules, if the molecular weight is less than 500 Daltons, and there are hydrogen bond donors are less than or equal to 5, or if the hydrogen bond acceptors are less than or equal to 10, and if this C log P is less than 5, now in C log P, P is the partition coefficient. Okay, it is defined as a partition of the drug in water or aqueous by the concentration in octanol. Okay, so if we partition the drug between these two and we find the concentration, and then we can calculate P like this. And if we take the log of that, and if it is less than 5, then Lipinski found that if a drug follows all these criteria, then it can be delivered per oral. It can be given orally. Okay, so the assumption by Lipinski, the criteria here is that these drugs can be delivered per oral and they will be absorbed by passive diffusion. Okay. So if this criteria is fulfilled, it can be absorbed by passive diffusion. Now, suppose there are drugs like, say, natural products, like any natural product drug. Okay. It will be observed that natural products strictly don't follow Lipinski's rule. Now there are so these are all, if we observe, multiples of 5. Okay. So this rule itself is called Lipinski's rule of 5. Okay, so some natural products don't follow Lipinski's rule or the Lipinski's rule of 5. So the question is why? Here, what is important to remember is the absorption is happening by passive diffusion when it follows Lipinski's rule. But if there is a natural product and if there is a transporter, if the natural product has got a transporter, then this rule does not apply, because this rule is applied only for drugs which are absorbed by passive diffusion. So the point is, drugs have experimentally been found to be drugs. So what makes a drug are certain criteria like this. What makes a drug a drug? Okay. So those are certain criteria like this which has been established experimentally. Okay, so because it has been established experimentally, we can accept it with a great deal of confidence. So why is this important? It is important because if we have this structure, then these properties can be calculated. Okay, what is the advantage if we calculate it? We can know beforehand, before the experiment, we can know, what can we know? We can know whether the drug can be given orally. Okay, whether it will be absorbed by passive diffusion. We can know the route. So if it cannot be given orally, then we can give it by the parenteral route. Okay, so if the molecule is very large and it does not follow a few of the Lipinski's rule, then we can think of optimizing it for parenteral administration. Okay. So suppose we have a large set of compounds. Okay. We can use this calculations to select the best candidate. Okay, for further development. Okay, shall we look at an example? Okay, for this, let's go back to the browser. Okay, let's type PubChem. Okay. Let's open up this and let's type in here aspirin. Okay. Let's click on that. So we get the 2D structure. Let's download this structure in an SDF format. Okay, so it is saved in the desktop. Let's go back to the search. Let's open up a new, let's say, Lipinski filter. Okay, we type in Lipinski filter and we get the site. Okay. So this is a site of the supercomputing facility for bioinformatics and computational biology at IIT Delhi. So we can choose the file that we saved. It is in desktop. We open the structure. Okay, so we have now put in the structure and we don't change the pH value and we submit. Okay. Right. So what did we do? We took this structure of aspirin and we want to check what is our goal here. We want to check whether it obeys the Lipinski's rule of 5. Can we give it orally? Okay, so we put the structure of the molecule. We got it from, where did we get it? We got it from PubChem, okay, in SDF format, and we pulled up this website and we put in the file and we submitted. See, it has calculated. It has calculated the mass to be 180. According to the Lipinski filter, it should be less than 500 Dalton, and this is 180 Dalton. The hydrogen bond donor, it should be less than 5 for it to be given orally. Here it is 1. And the hydrogen bond acceptor should be less than or equal to 10, and this is definitely less than 10, it is 4. And the log P value, the partition coefficient, it should be less than 5, and here it is 0.92. Okay. So what is the result of this analysis? We can say that aspirin is a drug that can be given orally. Okay, so this is the importance of knowing the filters. Are these the only filters available? No. Okay, if, in that same site we get this tool called drug likeness tool. Let's open up this. All right, so this tool can be used as an alternative to this site. But the good thing about this tool is that it has got more than one filter. Okay, if you observe, there is Lipinski's filter, there is MDDR rules, there is Weber rules, there is a Gauche filter, there is a BBB rule, CMC50 like rule, and there is a QED rule. So these are all drug filters, okay, that can be used for the analysis that we mentioned above. Okay, so let us transition back to. Okay, so in this lecture, we have done a small recapitulation based on what we did in one of the previous lectures. We talked about drugs, we have defined drugs, and here there are different definitions for drugs, but we have looked at the legal definition of drug in India. We looked at the broad mechanism of action of drugs. We said that drugs act on receptors, and those receptors, the most common receptors are proteins or nucleic acids. Then we went on to discuss the structural features of protein receptors. Then we broadly classified the drugs. We classified the drugs as small molecules and large molecules, or large molecules, which we call biopharmaceuticals or biological drugs. Then we talked about side effects of drugs. We said that the off-target effects are responsible for the side effects that are shown by drugs. Then we talked about some characteristics of small drug molecules, where we talked about some filters. We talked about the most important filter, which is Lipinski. And we looked at other drug filters. Okay. So with that,